Okay, good morning everybody. Thank you for the opportunity to give me the opportunity to talk to you today on tumor-educated monocytes. I'm actually the second speaker now in line with my previous colleague to talk about tumor stroma and not the tumor cells per se. We have heard a lot about genomic alterations in the tumor cells, but large parts of the tumor are actually formed from the stroma. And an important component of the stroma is the inflammatory cells. So we looked at this part of the tumor stroma to discover a genetic signature. Well, looking at the original purpose of the immune system, acute inflammation is the initial response of the body to harmful stimuli. So pathogens elicit an immune response and lead to extravasation of monocytes that differentiate into macrophages once they enter into the tissue. These macrophages, as part of the innate immune system, also interact with the adaptive immunity, the T cell and B cell immunity, and together with these orchestrate the immune response towards pathogenic stimuli. Now, chronic inflammation is quite different from acute inflammation in that it can promote actually tumor growth. In the initial phase, the proliferating cancer cells will, such as pathogens, elicit an anti-tumor response mediated by macrophages and the adaptive immunity. But once the tumor outgrows the immune response of the innate and adaptive immunity, you have the immunogenic switch, and the tumor hijacks the immune system for its own purposes, meaning that we have a pro-tumoral phenotype of the macrophages that will actually promote tumor growth and also inhibit the adaptive immune system and promote processes such as angiogenesis and further proliferation of the cancer cells. Now, we reasoned that also the precursor cells of the macrophages that are circulating in the bloodstream may be affected by the tumor cells already. And if so, could these cells be used to detect tumors? Well, we chose colorectal cancer, and I won't go through the details because we have heard this a couple of times already. One thing that I want to highlight, though, is that 1/3 of patients present with metastatic disease at the time point of diagnosis, and these advanced stages have a very poor prognosis. And out of those that are non-metastatic at presentation or diagnosis of the disease, about half will become metastatic during the course of the disease, rendering their prognosis worse. And current screening tools, we have discussed this a couple of times, like specificity or screening compliance, especially this is the case for colonoscopy. So blood-based gene marker panels are an ideal target for screening for colorectal cancer because it's an easy accessible source, it's non-invasive, and it might improve compliance for screening colonoscopy in those individuals that are flagged at high risk for colorectal cancer. This review is a little bit outdated and does not include the previous presentation, but most of these panels have sensitivities and specificities around 70 to 80%, so those can be improved, and our hypothesis-driven approach, in our view, had the opportunity to improve sensitivity and specificity. So once again, these monocytes, in our view, were an ideal target for screening for colorectal cancer, as they are the precursors of the more described macrophages that can either show pro-tumoral or anti-tumoral phenotypes, depending on the stromal context. I have to say with a bit of caution that a lot of the studies in the immune system have been done in mouse models, and actually the differences between mouse models and human immune system are quite marked. So especially for colorectal cancer, we have some data that describe the macrophages in the tumor, but those data are conflicting and probably depend on the stage of the disease and other factors. These peripheral blood monocytes we isolated in a two-step isolation process. First, a density gradient centrifugation that led to a peripheral blood mononuclear cell pellet, and from that we did a positive isolation for CD14, which is a pan-monocyte marker. In these pure monocytes that had a purity of above 90%, we did a genome-wide expression analysis to delineate a genetic profile. with differentially expressed genes that could be used as biomarkers. Our rationale was to take healthy volunteers without any evidence of disease and patients that had non-metastatic or metastatic disease at time point of diagnosis. In total, we included 370 patients in several cohorts. First, we had the gene panel identification by using the Illumina microarray assay to delineate the candidate biomarkers, and then we extensively validated this in a multicentric study with a different method, namely qPCR, and once analyzed here and finally delineated the genetic marker profile, we did a robustness assessment of this final panel of genes in another set of 133 patients comprising both colorectal cancer patients, healthy volunteers, but also other diseases to check for specificity of the assay. In first instance, we found 43 genes that were found to be differentially expressed between the overall cohort of patients and healthy volunteers. Most of them were upregulated, and quite strikingly, we have a really broad set of genes that did not show any differential expression at all. And you can see here in those that actually have a highly significant differential expression that the expression fold change is not several times, but it's between 1.5 and 2-fold at the maximum. So the expression changes are quite subtle, and we can discuss later on what might be reasons for this. Now, checking these in the different cohorts of metastatic patients and non-metastatic patients, we found a broad overlap of these genes and virtually no differential expression between metastatic patients and non-metastatic patients, indicating in our view that the gene changes in the circulating monocytes are imprinted at early onset of the disease and stay robust with disease progression. By doing a technical validation with an independent method, qPCR, we were able to confirm in total 23 genes to be differentially expressed. One of the reasons, and this was a question in the last talk, is probably that it is not the gene product itself, but it's also dependent on alternative splicing and the precise assay of qPCR that can detect the expression differences that may lead to different numbers of differentially expressed genes. We took this assay to a multicentric validation study and could show differential expression of all of these 23 genes as single genes for most of the cases. You can see here that virtually all of the genes are differentially expressed, but when taking single genes, we did this analysis in ROC analyses, the AUC, and the diagnostic accuracy was not good. So we did what also has been done in other assays and combined the informative value of the entire panel of genes in a multi-gene classifier with different approaches, and were able to have an exceedingly high diagnostic accuracy in this multicentric confirmation study with a sensitivity of up to 100% and a specificity of 93%. You can see here that we applied 3 different algorithms and then in the end an ensemble algorithm that combined the information from the other 3 as a majority vote. Now, quite obviously, this is a biased set of samples because we use the same set as a training and test set as had been done in several studies. So we took this defined gene panel to a completely independent set that included also screening patients, screening individuals negative to colonoscopy or positive to colonoscopy for colorectal cancer. And in this independent, also multicentric cohort, we were able to show a sensitivity of 92.6% with a specificity of 92.3%, so maintaining the good values that we had before. In wondering what led to these expression changes, we established an in vitro culture model where we were able to show for a subset of 7 out of these genes, differential expression in vitro that was specifically induced by soluble factors released from the transformed colon epithelium, as the expression of these genes was not altered in vitro by a benign colonic epithelial cell line, but solely for a malignant colon cell line. We performed these assays in normoxia and hypoxia to account for the fact that growing tumors have increasing hypoxic areas, and in line with the fact that the signature was imprinted at early disease onset and not changed over the course of disease, we did not see any change in expression depending on normoxic or hypoxic conditions. Now, monocytes are highly plastic cells and can change markedly depending on the microenvironment and also rapidly. So we were wondering if these expression changes would be reverted if we took the stimulus away. And indeed, we saw that taking away the medium that contained the soluble tumor-released factors, the expression levels went back to the expression levels of unstimulated monocytes. This prompted us to take this question to the in vivo situation in patients that had been curatively treated and were without any evidence of disease. And in these patients, up on curative treatment, we indeed saw a decrease of the expression levels of the previously elevated gene signature back to normal expression levels. Although this is a preliminary set containing only 15 patients, this is highly suggestive of the potential to use this signature for disease follow-up. Our data set contained only 4 patients that at the time of screening had presented with metachronous metastases and not with primary synchronous metastases, but these metachronously metastasized patients group very well in the cluster of colorectal cancer patients, indicating that the signature might also be useful to detect disease relapse in disease follow-up. We wondered if the expression changes that we saw were a general phenomenon of a malignant transformation or were specific to colorectal cancer. And to answer this question, in our in vitro model, we took a gastric cancer cell line and assessed the expression levels in comparison to the colorectal cancer cell line, and saw that the expression changes, with some exceptions, were not induced by a gastric cancer cell line. Logically, we took this question to the clinic and analyzed the signature in patients with gastroesophageal cancer or pancreatic cancer. And in both of these patient cohorts, we did not find any differences in expression compared to healthy volunteers, with the exception of these markers for gastric cancer. This is indicative in our view of a specific marker panel that can be delineated for several tumor entities, and there will be some kind of overlap. To detect this specific signature that might detect gastroesophageal or pancreatic cancer, we would need to do the genome-wide expression study on those patients again, which has not yet been done. Finally, we reasoned that the signature that we specifically found in monocytes might also be detectable in the entire fraction of mononuclear cells, which would make the assay much more easy to use in clinic because the procedure would be less laborious. However, checking this signature in the entire fraction of mononuclear cells did not show any differential expression, indicating the strength of the hypothesis-driven approach, but making the procedure of the diagnosis more laborious. This was not only the case for a single gene analysis, but also using the previously established classifier. In this independent cohort in monocytes, we found the previously confirmed sensitivity and specificity. Using this classifier in mononuclear cells, it was no better than guessing. So in total, overall, these cohorts, we were able to show a robust and impressive performance of the screening test with, as mentioned, about 92% sensitivity and specificity, which, depending on the precise cohort, ranged up to 100%. So, taking again the slide that I showed in the beginning, we are convinced that this hypothesis-driven approach has the high potential to increase the rates of sensitivity and specificity in non-invasive colorectal cancer screening. In conclusion, we found a strong diagnostic signature that is specific for colorectal cancer. It allows for non-invasive early detection. We have validated this clinically in a multi-center trial, and this signature of 23 genes that we found might actually be able to be narrowed down to a lower number to allow for a cheaper test. However, to not compromise our data set, we have not reduced this number of genes further, because this would have meant another round of robustness assessment with a newly defined signature. And this test shows a superior sensitivity and specificity relative to other non-invasive tests. Ongoing research questions are multiple. We need to explore if a smaller signature is equally capable of maintaining the diagnostic performance. We are working on a one-step monocyte isolation that makes the procedure less laborious. We are likewise working with a company in Belgium where most of the research was done to develop a commercial assay, including absolute standards that do not have to rely on a comparison to a defined cohort of healthy volunteers. As done in the pilot analysis, but now in a broader setting, we need to assess this test in an actual screening population, and this is done in Belgium, where there's a large screening effort to increase colonoscopy screening rates by rolling out FIT to all the population. And we work with the company to work together in this setting to compare directly to the FIT that is established in the clinic. We need to check the test performance in pre-malignant and benign polyps and see if there are differences, likewise in inflammatory conditions. And a big field of discussion is how treatment might affect the highly versatile monocytes and their expression behavior. This can be by anti-inflammatory medication or chemotherapy. This can be by treatment with radiation or by surgery, and these confounders might obviously influence the potential of the signature to be used for disease follow-up. And we need to assess the signature in disease progression, because the results that I showed you for a reversal of the signature or detection of metachronous metastases are preliminary. The possible applications thus are: once in follow-up of colorectal cancer patients after treatment, and this could mean that this test might be used in lower intervals and might also detect metastases that might be missed by colonoscopy. And it can obviously be applied in screening, either as a selection assay with a high sensitivity to flag people that need to undergo the screening colonoscopy, or if the scores are maintained, to screen an at-risk population that have been discussed mainly yesterday. This test reduces the unnecessary invasive, expensive testing, and it will increase screening compliance in comparison to colonoscopy. And you can very well discuss that this test has a higher specificity and sensitivity than any other existing assay. That does not include the recently in the New England Journal published fecal DNA test that also showed a superior sensitivity and test performance in comparison to most other assays. This work has been done in several cities. In my postdoc time in Leuven with Massimiliano Mazzone and Peter Carmelid, the statistician Walter von Delm in the VIB nucleomics core facility that helped in all the data analysis, my department here in Heidelberg with Markus Büchler, who is supporting me, and the cooperating institutions in Leuven, in Brussels, in Rome, and the people in the lab in Belgium that supported the entire study. And with this, I would like to open the discussion. Thank you very much.
Questions?
Hello. Do you know which is the soluble factor produced by the tumor cell, and would be possible to detect directly in the blood?
No, we have not checked which soluble factor this is. I skipped over it, obviously due to time limitations. We conditioned medium with tumor cells and did a filtration of that medium, so it will contain all the soluble factors from the tumor. We did this also with benign colon epithelium to exclude that it's general factors from conditioned mediums, such as acidosis, changes in lactate concentrations, and all these kinds of things. But the precise soluble factors are not determined within this study.
Okay, so I think at the end maybe it will be a more direct test if you can detect the soluble factor in the blood.
That would be correct, yes. Although, I mean, this is speculation, but it will not only be the soluble factors that are the only source, it will also be the interaction with the tumor stroma. At least this is the hypothesis behind this entire setting.
I was wondering whether I could still tease you further to speculate on mechanism, though.
Well, this is quite tough. I mean, obviously, we took this gene panel to ingenuity pathway analysis and tried to see if there is any pathways, any known described pathways behind it. However, monocytes in cancer have not been studied really a lot in literature, so it's quite hard to take on existing data, and we did not nail down any known pathway for this. However, there is an interesting overlap between this screening assay and an entirely different screening assay that was done in the same lab, where mouse macrophages were cultured with tumor-released soluble factors with or without hypoxia, and some of the markers independently popped up in both screenings. So this is suggestive that there is something going on in these cells. The difficult thing is that these monocytes are highly plastic, so any changes in the microenvironment will rapidly change also the expression levels of these genes. The M1, M2 phenotype that was originally described in describing macrophage heterogeneity is only extremes of an entire continuum of different expression phenotypes. And while this makes the assay appealing for disease monitoring, because it will revert to the healthy expression levels, it also makes it difficult to nail down these precise expression changes in a fixed assay.
I have one in addition to that. Does it work across species barriers then? So if you transplant human tissue into mouse? I may have missed that if you already indicated.
No, we have not done that. The problem with assessing tumor immunology in xenograft models is that virtually all xenograft models rely on immune deficient mice, and thus you will not pick up the changes in the macrophage line.
You mentioned that there wasn't any difference between, you know, when you get a metastasis, it doesn't change. Does it really not change at all? The microenvironment, basically, where
not within our data.
Right. So there's no optimism to be able to diagnose that particular.
No, no. And we also checked other things, and it was really an assay that picked up all colorectal cancer patients. For example, we checked left versus right colon cancer, rectal versus colon cancer, but there were no changes in that entire group of patients.
Further questions? Nobody vying for the microphone. Thank you very much. Due to organizational reasons, we have yet another coffee break. You go. But we're a bit early.